Students, welcome you all on the behalf of RAHUL ANAND tutorials. So as you know that I am RAHUL ANAND and this is somewhat around my channel whose name is actually the RAHUL ANAND tutorials. So now let's begin with the very interesting topics which are often being asked in terms of your Neet, often the Aiims, often the All India Pre-Medical Examination. So these are literally belonging from which of the chapters of your book. So these are belonging from the here, minute of the cell, the unit of life. So primarily when we are talking about these cases, so you have to understand the certain facts in it, such as what? So we are going to study about the fluid Mosaic model here. So fluid Mosaic model are actually what? So these are what? So these are the most popular, accepted, as well as the proposed result of what? So the most popular, accepted model of these in terms of the plasma membrane is actually the here minute of the fluid Mosaic model. Generally proposed by who? So it was proposed by the Singer as well as the Nicholson. In terms of what of the era? So it was around 1972. As well as when we are talking about this case, so you have to understand what. So in this case, what will be happening? So in this case, the protein bursts of what? So protein bursts of the ice block will particularly float in terms of what? In terms of the sea of the lipids. What will be happening? So in this case, the protein molecules are particularly being merged in terms of what? So your protein molecules are particularly being merged in terms of the bilayer of the lipid molecules. Often when we are talking about these, means what are actually the lipids? So you are very clear about that. So what are actually the lipids? So these are the natural occurring what? So these are the natural occurring substances which are particularly insoluble in terms of what? In terms of the water. Often when we are talking about the case as well as the role of these lipids, so playing an important role to provide what? To provide the strength mechanism with reference to what? With reference to our cell. Now, so let's proceed. So we are often having the different types of the components in terms of our cell. Remember? So we are having the nucleus, then the nucleoids, as well as these in terms of the centrioles. Often we are having the microchromatin network like projection, such as the cilia as well as the flagella. Different composition as well as the different types of the cell will be bearing these kind of the cilia as well as the flagella. As well as we are often having the two broad kind of the cells. So these are actually what? Tell me. So these are the here minute of the eukaryotic cell as well as the prokaryotic cell. So we'll be understanding about that in terms of our next lecture. Now, so when we are talking about these cases in terms of what? In terms of the fluid Mosaic model. So you have to understand the protein case in brief actually. So we are having actually here how many types of the protein? Tell me. So here we are having the two broad kinds of the protein, such as what? Such as the integral protein as well as what? As well as the peripheral protein. So thus when we are talking about these cases in terms of the protein, so you have to understand the certain names as well as the protein types in it. So we are having what kind of the protein? So here we are having the integral protein, then the peripheral protein. So when we are talking about these cases, meaning of the integral protein, so it will be present where? So it will be either merged as well as what? So it will be either merged as well as present in terms of the here, mean on the membrane. But when we are caring as well as understanding about the second case, in terms of what? In terms of the peripheral. So by the name you are very clear, means what? So the peri actually means what? So this will be present in terms of what? On the surface of actually the membrane or either in the surface of the membrane. After that you are often having the third composition in terms of our protein. So these are actually what? So these are the globular protein. Remember? So we are having the here minute of the fibrous protein, often the globular protein. So globular protein are actually what? So these are quite a bit soluble in terms of the water molecules. Thus, these are often being found in terms of what of the reason in terms of our abdominal cavity. So when we are talking about the case of the here minute of the globular protein, so the best example with reference to it will be actually what? So these are the pepsin, often the renin as well as the lipids. So these are actually coming under what case? So these are actually coming under your here minute of the globular protein. Playing important role. Means what? These lipids, often the renin, in order for what? In order for the digestion as well as the coagulation of the milk, as well as playing important role to synthesize the organic components, as well as freeing it towards what? As well as freeing it in order to acquire the sufficient bulky amount of the energy. Understood? So when we are talking about these cases in terms of the integral protein as well as the peripheral protein, so you could understand that. So this will be particularly present in terms of what? So this will be either present in terms of the membrane space. But when we are talking about the case of this minute of the globular protein, so it will be present where? So your actually globular protein will be particularly present, but it will be present where? So it will be present in terms of the separate ratio as well as the proportion in terms of what? In terms of your fluid Mosaic model. So now often when we are talking about this case in terms of the integral protein as well as the fibrous protein, so in this case what will be happening? So these are the highly viscous fluid which will be present in terms of the membrane of what? Of the phospholipid bilayer. So you are very clear about that. Thus, when we are talking about this case in terms of the phospholipid molecular level, so in this case what will be happening? So this will play an important role to provide the barrier against what? So this will be playing an important role to provide the barrier against the water soluble substances as well as the water molecule. So you are very clear about that, means what are actually the water soluble substances, often the H2O or what. Now, so let's proceed. Thus, when we are talking about this case in terms of what? In terms of the phospholipid molecular level. So in this case, what will be happening? So this will provide what? So this will provide the relative impermeable barrier against what? Against the water soluble substances, often the water molecules. So you are very clear about that, means what are actually the molecules as well as the here many of the water soluble molecules. Often what? Often the H2. Now, so let's proceed. So when we are talking about the fluid Mosaic model, so you have to understand the certain facts in it. So when we are talking about the particles arrangement in terms of these model, means what? In terms of the fluid Mosaic model. So there will be particularly arrangement in terms of what pattern? So it will be particularly arranged in terms of the Mosaic pattern. So Mosaic is particularly the type of arrangement often. So this will be present in terms of what? So it will be actually present in terms of the both side of the phospholipid bilayer. So you are very clear about that, means what are actually the phospholipid bilayer as well as its presence will be in what region. So find here. So here is the phospholipid bilayer. Often your phospholipid bilayer as well as what? As well as the fluid Mosaic model is having the different types of the components as well as the composition in it. So these are actually what? So we'll be often having the cholesterol as well as what? As well as the integral protein. Then what? Then the peripheral protein. Often the bit amount of the globular protein. Then what? So often we'll be having the sugar. Then the cholesterol as well as what? As well as the carbohydrate. So remember about the case of the carbohydrate. Often when we are talking about these cases, means what are actually the carbohydrates. So you are very clear about that. So these are often being further divided in terms of the three broad categories. So these are what? So these are the monosaccharide, then what? Then the polysaccharide, then the oligosaccharide. So now let's understand about this. So monosaccharide are directly proportional for what? So the carbohydrate which could be broken down into what? So the carbohydrate which could be broken down into 2 to 10 units of the simpler sugar. Coming under what case? So it will be coming under your monosaccharide case. After that you are having the here when in terms of the polysaccharide. So the carbohydrate which could be broken down into what? Into the more than 10 units. It will be coming under what case? So it will be coming under your, tell me, here when in terms of the polysaccharide. After that in last you are having the oligosaccharide. So the carbohydrate which could be more broken down in terms of what? Into more than, particularly here in terms of 15 to more units. So it will be coming under what case? So it will be coming under your oligosaccharides. Remember? So poly as well as what? As well as the mono. Now after that you are often having the different types of the things in it, such as what? Such as the protein. So here we have dealt about the protein in terms of the integral protein as well as the peripheral protein as well as what? As well as the sugar. So now let's understand about this in terms of the brief. So when we are talking about the presence in terms of these risks, in terms of the fluid Mosaic model, so we'll be having the two types of the protein, of course. So these are what? So these are actually the intrinsic protein. Often what? Often the extrinsic protein. So when we are talking about the case of the extrinsic protein, so by the name itself it is very clear, of course. So extrinsic means what? So something which will be present in terms of what? In terms of the inferior ratio, as well as whose existence as well as particularly going potential is literally what? Is higher as compared to the intrinsic protein. Thus this will be coming under what case? Thus these are particularly given the specialized names. So these are known as what? So these are known as the extrinsic protein. Often when we are talking about the ratio in terms of these rates of what? In terms of the fluid Mosaic model. So it will be around how much? So it will be around here in terms of the 30% of the protein. So these are in terms of the 30% of the protein are what? So these are the extrinsic protein. Thus here around 30% of the total protein will be occupied by what? So it will be occupied with reference to this mean in terms of the loosely packed, often at what is at the polar surface of the liquid bipolar. So these are particularly known as what? So these are known as the extrinsic protein. So you are often very clear about that, means when we are talking about these, means what actually? Means the extrinsic protein. So it will be what? So it will be loosely packed. Thus in this case what will be happening? So in this case the escaping mechanism will be very easier. So you would often find it on the board. Find it. So the escaping mechanism with reference from what? From the lipid bipolar. Often when we are talking about these cases, means what are actually the lipid bipolar. So you are very clear about that. So remember about the lipids. So what are actually this? So bipolar actually means what? Or either the bilayer actually means what? So it means the particular layer which will be playing important role as well as present in order of what? In order of the mechanism of these what? The polar surface of this mean term of the fluid Mosaic model region. Often when we are talking about the lipids, so these are actually what? Tell me. So these are the natural occurring what? Organic substances which are particularly insoluble in the water molecules, as well as when we are talking about these mechanisms, what are actually helpful. So these are particularly helpful in order for what? So these are particularly helpful in order to provide a particular here structure as well as the membrane as well as the strength to what actually? To our cells. So as well as when we are talking about the another case, so you are often having what? So you are often having the intrinsic protein. When we are talking about the intrinsic protein case, so the case will be quite a bit different. Means what? So in the case of the intrinsic protein, what? As well as when we are talking about the case in terms of what? In terms of the intrinsic protein, so around how much of the protein will be particularly merged down or either penetrated with reference to what? With reference to the lipid bilayer, so it will be around 70%. Remember? So on the 30% directly proportional for the extrinsic protein. Often when we are talking about these cases in terms of the 70%, so it will be employed with reference to what? With reference to the intrinsic protein. So these will be particularly merged down with reference to what? In order of the here, meaning in order for the lipid bilayer. Often you are very clear about that, means what are actually the lipid, not need to explain you about that. And after that, what will be happening? Thus, the particular escaping mechanism with reference to what? With reference to this in order from the lipid bipolar will be particularly difficult. So when we are talking about these cases, so you have to remember the certain more parts in it. So when we are talking about the here in order for the intrinsic protein, so you could often find here on the board in order from the here. Thus, when we are talking about the intrinsic protein, so you could understand that these are just like what? So these are just like the icebergs floating in the sea of the lipid. So you are often very clear. So you could often find here on the board. So you are having the fluid Mosaic model. And often in these cases on the middle structure you are having what? So you are having the integral protein. Find here. So this is somewhat the portion on which what? Your integral protein will be particularly. As well as when we are talking about these cases in order of what? In order of the globular protein. So your globular protein will be playing an important role as like what? As like the here in order of the enzyme carrier. So often you are very clear about that, which what are actually the globular protein as well as the enzymes. So now let's understand what are actually the globular protein. So these will be what? So these are particularly soluble in order of the water molecules. Often the best example will be what? So these are actually the renin as well as the pepsin. So these are generally present where in order of the stomach or either the abdominal cavity, playing important role in order to carry out the course of what? In order to carry out the course of the digestion. Often when we are talking about the case of the enzymes, so you are very clearly understanding what are actually the enzymes. Tell me. So enzymes are the particular catalyst playing important role for what? Playing important role to increase or rather decrease the rate of reaction without particularly being what? So without particularly being altered. So this is the condition when we are talking about the case of the enzymes. Often your enzymes are particularly being divided in order of the two broad categories. So these are what? So these are the coenzymes, often the apo-enzymes or rather the apopectins. The broad categories with reference to what? With reference to the enzymes. In the one, we will be having the protein, and on the other, you are having the absence of the protein. Thus, specialized in order with reference to the two names. Means what? In order of the cofactors as well as the apo-enzymes. So this is somewhat the condition when we are talking about the fluid Mosaic model. In which we have understood about the protein. So your fluid Mosaic model are often known as what? So they are known as the protein Mosaic model. In which we have understood about the bit introduction of what are actually the integral protein as well as what? As well as the peripheral protein. Often you are having the certain more parts such as what? Such as the carbohydrate as well as the cholesterol as well as these things in order of the sugar. Often in order for the globular protein. So we will be understanding about that in terms of our next lecture. Otherwise you will be not be able to get that. So you could often write these. So if you like the work with reference to the education as well as the lectures as well as what? As well as the videos. So you could often subscribe, like as well as comment below in the text. Thus I will be knowing that what concept actually you are hungry for as well as what concept you haven't understood or either got. So very thank you. With reference to what? With reference to the RAHUL ANAND tutorials. So very thank you.